I'd like to welcome you guys to Unit 2 with focus here is energy, and our theme for this unit is conserved core processes. Now you guys know that conserved core processes are going to be processes that we're going to see across multiple organisms, different species throughout the living universe. Right. So here we have a E. coli bacteria, we have a beetle, we have a redwood, we have a jellyfish, we have fungus, a mushroom, and then we have an amoeba. Now, so what do you think is a process that they all share in common? You ready? The answer is glycolysis. All right, and glycolysis is a 10-step pathway that starts the breakdown of glucose and is anaerobic. It yields us 2 ATP for 1 glucose molecule. It is not super efficient, but every single organism that we know of is going to carry out some form of glycolysis. Now, well, I can't say every single organism, but there's only a few minor exceptions to ones that don't. So it's a very widely spread out process. Now, what this allows us to do is to look for patterns that are going to be similar between organisms that look wildly different. I mean, we're not ever going to say a bacteria is like a beetle, but these organisms look wildly different, and yet they carry out processes that are shared. So this again gives us evidence for common ancestry. That means we shared a common ancestor. So even though we don't look like bacteria, if we share a process with bacteria, that means that at some point we had a common ancestor. So conserved core processes and common ancestry both go in common, or hand in hand. Now, we've been talking about enzymes a little bit. We introduced this already in class. I just want to make sure you guys understood that enzymes are going to be a type of protein. They are globular in shape, like we see up here, and that enzymes are going to be building up or breaking down molecules. In the case that we have here, transforming the molecules even, in the case we have here, the enzyme is going to take 2 substrates and it's going to change those substrates and it's going to release products. And then that enzyme is going to go repeat that function again and again and again and again. And remember that enzymes are going to be speeding up the reaction rate vastly faster than these things would happen randomly. And here's an example of one that's just going to be using transferring. It's called substrate level phosphorylation. It takes a phosphate from a substrate and it transfers it to an ADP molecule to form ATP, and also releases a product. So the enzyme is not changed by the reaction, and it changes the substrates. And remember, enzymes do have the active site, which is where the interaction is going to take place. And we have something there called induced fit. So when Ben and I shook hands and we induced the fit around there, it's not like a lock and key model, but it's an induced fit. So when the enzyme binds the substrate, it's going to cause a change in the shape of the substrate that's going to make the reaction more likely to occur. Now, there's lots of factors that affect enzyme activity. And the 2 most common ones that you guys are going to look at are going to be temperature and pH. So we can kind of see here with temperature that we have this slow buildup in the rate of reaction as we move towards what's called an ideal temperature, and then we see a fast drop off after that. And the question you have to ask is, why is that going to occur? And the reason for that is that prior to the beginning here in the early stages, as it's colder, the enzyme is simply not moving as fast. It's not running to any of these substrates. Where it is, it's not running into them at a very high rate, so its reaction rate is very slow. But as we move towards the peak, though, it actually has its peak activity. But once we get very hot, then what happens is the energy from the water, or the fluid, is going to be causing disruptions in the shape of the enzyme, and then the substrate can't fit in the active site, and then the activity of the enzyme is going to decline. And now these disruptions typically are permanent. So if you heat or boil a enzyme, you're going to denature it. And denature means it loses its shape completely. Now, the other thing that can be is it can be an optimal pH, and we know that most organisms have a pH around neutral. Even in our animals, we're going to have a gut, and in the gut you're going to have stomach, and the stomach is very acidic. So some enzymes like pepsin only work at a very low pH. Like this one here, pepsin only works at a pH of 2. So if you took this pepsin out of the stomach and put it in the small intestine, the pH is much higher. It's going to denature. So where pepsin would denature at a very, very low pH, or very neutral pH, closer to neutral pH, in both cases the interaction between the pH level of the fluid and the enzyme is going to cause permanent change in the shape of the enzyme. And so either side of the pH is going to be denatured. Whereas it only gets denatured on the temperature one on the high side. And we also have substrate concentration. Sometimes you have just enough enzyme to do the job, and you can maximize the activity of the enzyme up to a certain point. And then if you've just got way more substrate than you've got enzyme, all the active sites in the enzymes are being filled maximally, and so we're never going to get a rate higher than this right here. That's why it levels off. So as substrate concentration increases, we will get an exponential curve, but then we are going to have it level off when all the active sites will be bound there, and the enzyme can't go any faster than that, can't work any faster. Of course, the other thing you could do is you could increase the enzyme concentration, and as you do that, your activity could continue to go up for very long periods of time. Because remember, when you think about enzyme and substrate, we always show you one picture, but it could be that we are having you there's thousands of enzymes in there, and the faster a process is, the more enzymes that are involved in it. So thousands of copies of the same enzyme. Now, a couple other facts we already talked a little bit about in class is that enzymes never work alone or rarely work alone. We're actually going to demo an enzyme that works alone in our enzyme catalysis lab, but it really doesn't work alone. We're just kind of pulling it out to demo it for you. And then they're going to work in series, where the product of one is the substrate for the next. And these series are called metabolic pathways. Those are those conserved core processes we talked about. And we have lots and lots of them in living things. Here is a general map of most of the reactions that are occurring in living things, metabolically speaking. And then we zoom in on this, I think this is P4 on this map right here. We can see that we have citrate right here, so we're starting the citric acid cycle, which we'll mention here in a few minutes, and then some of the other processes. And so you can kind of see here in the zoom in right there, you've got an enzyme there, got an enzyme there. So all these metabolic pathways are controlled by these little blue lettered enzymes, and they're going to be allowing us to adapt to many different kinds of situations. So when you eat food, the food is not just made out of glucose, it's made out of lots of different kinds of organic macromolecules, and your body can process pretty much all of them. So there is lots of stuff here that we can explore. If you wanted to look at metabolism and all the factors from it, you probably spend an entire career. Most people specialize in one area. So we said that we wanted to use metabolic pathways as our example here, and we said that defects in metabolic pathways were the result of defects in enzymes. And defects in enzymes then allow or cause these disorders, and such as PKU. So if you have a PKU sufferer, they have one of their enzymes, which is called PAH, and that is going to be taking one of the steps in phenylalanine, the breakdown to what's called I can't remember what's called off the top of my head, but it's going to be breaking down to that final thing. And if you look over here, what you're going to see is it's the first step, it's the first enzyme that eventually leads to the production of thyroxine, which is important for metabolic rate in animals, and then melanin, which is important for color. So if you have PKU, this enzyme doesn't work, you're going to have trouble producing thyroxine, and you're not going to be able to produce any melanin. And so therefore they're going to be fair, if it's for humans, they would be fair-skinned, and they would have trouble mediating their body temperature, because thyroxine, that's its primary job. So an enzyme with one enzyme defect is going to cause this entire cascade of events to occur. DOPA is a neurotransmitter, and that is going to be have brain effects. So some of the other symptoms that you would see from PKU might include behavior problems or brain damage. So just one enzyme in a pathway that's defective can lead to what's called a metabolic disorder. And then we have lots of examples of that. You just need to know PKU. All right, so you know that we need energy from the outside, and for heterotrophs, that energy is coming from food, and for autotrophs, that energy is coming from the sun. And so here's a photosynthetic food chain, and so the chloroplast is going to capture that light energy, and then they're going to use that to those plants are going to use it to build their tissue, then organisms are going to eat those plants, and those plants are going to get the energy from there, and then organisms are going to eat those, right? In a chemosynthetic pathway, we are getting energy, and not from the sun or not from food, but we are getting it from a chemical sulfide, and these are happening at the hydrothermal vents, which are found at the ocean bottom, where molten lava is coming up from underneath. So there's cracks in the surface of the plate. The lava is going to erupt from underneath, and that's going to produce this sulfide. And then we have these giant tube worms and some other crazy bacteria that are down there that are fixing that sulfur and using that as their energy source. And then they're going to have stuff that eats that, or bacteria that produce that, and then there are going to be animals that eat those things. So we have constant input of free energy, and we have this totally cool system that can happen at the plates, or at the tectonic plates where there's cracks in there. And it could be, a lot of scientists think that this is where life originally occurred, not at the surface of the ocean, but actually in the depths of the ocean. So it wasn't sunlight that was the first thing, it was these hydrothermal vents that were the source of it. So the energy that you're going to get in a typical reaction, we're going to have an exergonic reaction where we're going to be breaking down food, and then that is going to be captured in energy in ATP, and then that is going to be used for endergonic reactions, which will be then using that energy to make stuff or to do some kind of activity. So we have this path or pairing between exergonic reactions and endergonic reactions. It's not a direct one-to-one. The transfer is going to be ATP. So it could be captured by ATP, stored for several hours, and then used. It's not like, oh, I have to have lunch today, let me eat some food, and then I can do work. But if I don't have this food, then I'm not going to be able to do any work at all. You have plenty of energy stored from other areas. ATP can be converted from other sources. It's not necessarily a direct one-to-one relationship, but it is a pairing between exergonic reactions and endergonic reactions. All right, so the next thing, it was cycles, and we have two metabolic cycles that we're going to focus on. One of those is called the citric acid cycle, and you can kind of see here. I'll kind of blow that up for you a little bit and bring it forward. The citric acid cycle is going to be Citric acid cycle is going to be one where we have 1, 2, 3, 4, 5, 6, 7, 8 enzymes involved, and they are going to be taking an acetyl group, which is just a 2-carbon chain. It's if you started with 6 carbons in glucose, we're going to break it down to 2 3-carbon molecules, and then we're going to steal one of the carbons from that. So you have a 3-carbon chain, and that's the acetyl group, and then that is brought in to pair with this one enzyme called this one molecule called oxaloacetate, and those two put together to form citrate, which is how we got the citric acid cycle. And you can see at every step here, something is added, something is taken away, and we have something being oxidized and something being reduced. Now, if you remember oxidation is where a molecule is going to be losing either electrons or hydrogen or both, and then something that's going to be reduced is going to be gaining those things. So if you look over here, the NAD is being reduced to NADH, and over here you see that the FAD is being reduced to FADH. So the molecule here, succinate to fumarate, that is being oxidized, and then this one's being reduced. So just like we had up here, a pairing of endergonic and exergonic reactions, exergonic and endergonic reactions, that is what's happening specifically here. From this molecule to this molecule, we have a loss of electrons and hydrogens. Those are being that molecule has been oxidized, and then we have this molecule is transferred to atoms or protons transferred to this other molecule, so it gets reduced. So oxidized, reduced, oxidized, reduced. So every time you see this, that's getting reduced, then that means that this molecule is being oxidized. So we see the same thing we saw with enzymes before. They're transforming molecules, transferring stuff from one thing to another. The enzymes themselves are not changed by the reaction. Now, metabolic rate is something that we're going to see a fair bit in this one that we haven't seen too much in the past, but you can kind of estimate what's going to happen here is that you can have some organisms, the smaller they are, the higher their metabolic rates are going to be. And so, their heart rate would be faster, they would need more energy per unit mass, so a harvest mouse compared to an elephant would be much higher in terms of its energetic demands relative to its body size. So it's not that elephants are cold-blooded or anything like that. It's just that when the elephants are, if we compare their metabolism, it's much slower per unit body mass than the harvest mouse. And so we get this kind of nice little curve here, and you kind of see humans, we're kind of on the low end here. All right. Now, mitochondria, we just showed you the citric acid cycle, and the citric acid cycle is a catabolic reaction because we start with a 2-carbon molecule and we end up getting all of that carbon joined to form carbon dioxide, all right? And then we're going to form some water, and we're going to form some other pieces. Basically, this right here is going to get disassembled. It's going to be broken down. That's the catabolic reaction. All right, so mitochondria are going to carry out catabolism, and on the other side, chloroplasts, they're going to carry out anabolic reactions. They're going to be building stuff. So this is the Calvin cycle, named for Melvin Calvin, the guy who discovered this thing, and probably wasn't just him, but his whole crew. And what's going to happen is that this, slide that up there a little bit, this carbon dioxide is going to come in. They're one carbon molecules, and then through a series of reactions, and you kind of see these reactions here again. We got, if we get here to here, that would be one enzyme. Here to here, that's second enzyme, third enzyme, fourth enzyme, fifth, sixth, seventh, eighth enzymes, 8 or 9 enzymes. I'm not exactly sure, but you're going to form this larger molecule. So you start with a 1 carbon molecule, and then you end up with a 3 carbon molecule. Take those 2 3 carbon molecules, put them together, and you got yourself a 6 carbon molecule. You could call it glucose. So we always hear that chloroplasts are making glucose. It's not the only thing they make. They're making these 3 carbon molecules here. Glyceraldehyde, phosphate, and try that again. Control and 1. So the glyceraldehyde phosphate, the G3P right here, is going to be the molecule that then can go be used to assemble glucose and other organic compounds. Anything you can think of, the plant can make. They are a molecule making facility, and we are all completely dependent on them because they are getting us all the molecules that we need. If their plants aren't there, we're not getting the molecules we need. All right, so these are 2 metabolic pathways. One is going to be catabolic, breaking down. One's going to be anabolic, and that's building up. One occurs in the mitochondria, and the other occurs in the chloroplast. All right, so now we're going to capture and store energy, and you're going to see here that we saw the citric acid cycle in the last one, saw the Calvin cycle there. We're going to see this molecule right here. So if we start out with glucose over here, we end up with carbon dioxide coming out of that, and then all of the pieces of that glucose are going to get broken down, and all the hydrogen in the glucose, remember the glucose is a C6H12O6 molecule, and the glucose, the hydrogens, those 12 hydrogens, are going to end up making NADH molecules. And the NADH molecules are going to be transferred into the inner membrane of the mitochondria right here, and then they are going to donate those electrons and high protons to what's called the electron transport chain. It's going to pump them from the inner to the outer section, and then because they're going to create at a very high concentration here, a pump, remember, means that we're moving from a high concentration to a low concentration, or from a low concentration to a high concentration. They're going to be pumped up here. They're going to want to come back. When they diffuse through this enzyme that's called ATP synthase. That's the baby that makes ATP. So all these processes right here are going to be breaking down the glucose and then making a transfer molecule that is called NADH and FADH, and then those molecules donate their electrons and protons to this electron transport chain, and that is what we use to create ATP. Now the same thing happens over here in the light reactions in photosynthesis. So in the light reactions of photosynthesis, we have electron transport, and the structure is exactly the same. The only thing that's different is the method by which we capture the light energy. And the light energy, like you learned in the last unit, is coming from chlorophyll. So the chlorophyll is capturing the light energy, and it's using that to... My dog is trying to get my attention here. Sorry, pup, you're going to have to wait. So the energy of these reactions is going to be, again, transferred to this electron transport chain, and then that is going to be used to pump protons from the thylakoid, from the stroma to the thylakoid space, and then from the thylakoid space we're going to pump back out here. So we have the same process going on in 2 different organelles. Now the source of energy is different. The source of energy over here is glucose, and the source of energy over here is light, but it's capturing it, and they're going to store the energy in ATP, and then we can use the energy to do work in the mitochondria. That's going to pump it out of there. It's a mitochondria, the ATP factory, and the chloroplast is going to use that to help make our glucose, which then is going to go over here. All right. So if you have this electron transport chain, then you have a, wait for it, conserved core process. So conserved core process is going to be the last thing we'll talk about again that we started with here. Electron transport, ATP synthase are universal molecules that you find in all mitochondria and chloroplasts. So that must mean that they had at some point some kind of relationship with each other. Again, the conserved core process is evidence of common ancestry. So that's it for energy. Good luck, and we will see you in class.